Hi, I'm Vinita.
Hi, I'm Shumon.
And I'm Scott Gilbert.
We have a very special guest with us today here. Scott F. Gilbert is the Howard A. Schneiderman Professor of Biology Emeritus at Swarthmore College, where he teaches developmental genetics, embryology, and the history and critics of biology. He's also a Finland Distinguished Professor Emeritus at the University of Helsinki. He received his BA in both biology and religion from Wesleyan University, and he earned his PhD in biology from the Pediatric Genetics Laboratory of Dr. Barbara Meegan at Johns Hopkins University.
He did his MA in History of Sciences from the Johns Hopkins University under the supervision of Dr. Donna Harvey. He pursued postdoctoral research at University of Wisconsin in the laboratories of Dr. Masayu Omura and Dr. Robert Orbach. He is author of 3 textbooks, developmental biology being one of the most read textbooks and hailed as the Bible of developmental biology by students across the globe. Ecological Developmental Biology, Fear, Wonder, and Sciences in the New Ages of Reproductive Biotechnology. We would request you to talk about your work.
The questions that really excite me are questions that link development and evolution. And so the first big question that I wanted to look at was, how do changes in development cause changes in evolution? How do you, I wasn't so much concerned with the survival of the fittest as the arrival of the fittest. How do you get new variations that survive? And so I chose to look at one of the most beautiful and odd animals ever, the turtle. And I wanted to know, how does the turtle get its shell? And there are 2 parts of the shell. There's the upper shell, the carapace, and the bottom shell, the plastron, and they fit together, but they're made independently. And I wanted to know, how was it that you could get a new structure? How can you get a new structure without killing the old structure? And so we looked at the turtle shell, and we found that the thing that made a turtle were its ribs. The ribs went the wrong way. Instead of going ventrally to form a rib cage, the turtle ribs went dorsally and laterally. They went out from the vertebra into the dermis of the skin. And so we tried to find out how it was that the turtle's ribs were being redirected. And we found that it was due to fibroblast growth factors. Then once the turtle ribs were in the dermis of the skin, we wanted to know how it made the shell from the ribs. And we found that each rib acted as a signaling center and produced bone morphogenesis proteins, which went out from the rib and turned the dermis into bone. So the top shell was formed by an FGF signal getting the ribs into the skin, and then a BMP signal from the ribs. And the plastron of the turtle looks like, we're still trying to get hard data on this, but it looks like the plastron of the turtle, the lower shell, is made by neural crest cells, the same cells that form our face. So we're looking at that. The second question that I wanted to know was, in addition to the gene changes that can create evolution by changing development, are there changes that symbionts can bring to change development, and in so give new possibilities for evolution? And here we're doing research primarily on the computer on how do animals become herbivores? How do they become eaters of plants? Because interestingly, the first animals were probably detritivores eating, kind of decayed matter. But then you had carnivores. Herbivores came after carnivores. Usually one thinks of detritivores, then the herbivores, then the carnivores, but the herbivores came last. And they came from carnivores that learned how to eat plants. Now, how do you learn how to eat a plant? Because there's no genome in a vertebrate that allows us to eat plants. That's all done by bacteria and fungi in our guts. And so what interests me right now is the evolution of herbivory in animals like the cow, because the cow has a specialized region of the stomach, the rumen, which is used for eating grain and grass. But the cow is not born with a functional rumen. And here's where it gets interesting. The rumen not only houses the bacteria that digest the grass, the bacteria in the rumen actually build the rumen. Before the cow eats grass or grains, the rumen is rudimentary and it's not functional. But when the bacteria that's already in that area metabolize the grains and grasses, they make butyric acid. And butyric acid induces the rumen to form. So the animal isn't born with a functional rumen. The bacteria make it a functional rumen. And so I'm looking at how development can change when you get symbionts, such as the symbiotic bacteria, and how that leads to whole new paths of evolution, such as the herbivores, the cows, the ruminants, the giraffes, the rhinos. These are very successful animals, and they're successful because they learned how to house their bacteria. I just find that fascinating. I think the whole concept of becoming, the whole concept of cyclical renewal, the whole notion of how do you change but stay the same?
Exactly.
What I think is so important to remember is that all these discoveries about the brain microbe axis, these were made after 2000. This is a new science. This is a science which hasn't really gelled yet. It's still in formation. And I think that's very exciting because it means that people can go into this science, pretty much as it's beginning. And I think that this integration of developmental biology and microbiology and applied ecology are making a totally new way of looking at the world. And the whole notion of what is an individual, what is thinking, what is the immune system are all changing. And so what I learned in college, I have had to unlearn.
You have been in science for like a long time now. And was there any incident you can recollect that made you fall in love with science? When did you start falling in love with science?
Oh, I can't remember a time when I had not been in love with nature or science. I was a person who had the good fortune of being in a place during the summer which was full of frogs, salamanders, and butterflies. And whereas my friends knew all the baseball players, I knew all the butterflies. I knew all the names of the salamanders. Actually, I had it easy because the names of the baseball players changed. But that's who I always loved nature. And I think one of the interesting things, if I was to say that butterflies and frogs were the two things that I really liked, and both of them undergo metamorphosis in a very big way. And so maybe that pushed me for developmental biology. But I've always been interested in metamorphosis and nature. Yeah.
So does your childhood actually affect the cover of the book?
Uh,
A few additions.
the cover of the book is a very complicated story because we want a photograph that's striking, that's a beautiful
Yeah.
image. We also wanted to display a technique that is new and is allowing us to see something we hadn't seen before. And then the third thing is that we want it to be an organism that is interesting to look at. And we also want it, if possible, to be an entire organism and not a piece of the organism.
Yeah.
We only went to a piece of the organism once. Usually it's a whole organism, but I saw some pictures of the neural tube and I said, oh, I have to use these pictures.
Yeah.
Yeah.
So science and religion are often seen as two opposite poles, and you did your BA degree in biology and religion. So how did it shape your views as a graduate student?
Oh, that's a very interesting question, and actually for my life, a very important question. Oh, where? Okay, first, let me discuss the relationship of science and religion. I think that they are cousins, that they're not apart. They are family. And here's why. I think that the primary response of the mind to the outside world is wonder. That you have this experience of wonder. Now, wonder comes in two flavors. There's I wonder, and there's wonder. You know, the same word is used for both. And I think you have wonder. Wonder has a short half-life. You can't always be in a state of wonder without being a mystic. So wonder becomes curiosity, and wonder becomes awe. And from curiosity, you get science and you get philosophy. From awe, you get reverence and religion. And so I see science and religion as kind of grandchildren of wonder. And for embryologists, there's never a big separation between curiosity and awe. That when you see a chick 3-day embryo and you see the heart begin to beat, you know, and you see the neural tube forming, and I've seen that hundreds of times. I am still amazed by it. So I think that awe actually inspires us to do more science, which is the curiosity. So I don't think that there's a separation. Now, of course, if you want to talk theology, kind of the science of religion, then you get disputes because you have two things that both say we're knowledge-based coming into conflict. But I don't think science as science and religion as religion, I think they're within the same family.
That's the most beautiful explanation
Wow.
that I've ever heard.
For me personally, it was very interesting because it got me as a evolutionary developmental biologist in America. I have to deal with creationists who don't like evolution. I also have to deal with the abortion debate, which is about embryology. And I have to concern myself and I have to speak with people who are coming from a religion background. And I can do this because I know the vocabulary. And so I've been invited to go to conferences in the Vatican. They didn't listen to me, but I also went in 2016 to visit the Dalai Lama and to talk about embryology to the Dalai Lama. Now, the Dalai Lama has said, you probably know this, that if
Yeah.
he were not the Dalai Lama, he wants to be
Yeah.
a neurobiologist.
Yeah.
Yeah, I saw that. It was an amazing.
And what he said to me after my talk, he said, well, first he said he liked the talk, which made me very happy. But then he said, the questions that interest him are how the reptile brain becomes the mammal brain and how the mammal brain becomes the human brain. So I figure he's interested in evolutionary developmental neurobiology.
Developmental biology.
Exactly.
Mentorship plays a very big role in academia as science progresses through passage of great ideas, and whatever we discover today is because we are already standing on shoulders of giants. So therefore, how students are mentored does shape the progress of the whole idea of doing science. What is your idea and mantra of mentorship, since you must have been mentoring a lot of students in like all these decades?
Well, I've been incredibly lucky because I've both been mentoring students and I have been mentored by some remarkable people. And when I was in high school, there was this doctor who saw that I was interested in biology, and he invited me to his laboratory in New York City. And so
Wow.
I started just working in someone's laboratory as a high school student. You know, I was just really watching, but that was fascinating. And then I went again as a high school student to a course that was run by the National Science Foundation. And it was a course to teach high school students real biology. You know, it was saying that, you know, in the high schools, you don't get talked about DNA, and you don't get talked about cell biology, and you don't get talked about what's happening in paleontology. And so I went to this course for 6 weeks, and I don't think I was ever so excited to be in any one place, because here were all these other high school students who were hungry to talk about biology. And here was this professor who looked down on us and says, "You high school students, you don't know anything about biology. And I am going to show you how little you know about biology, because I am going to give you so much homework and reading that you will have to stay up nights to come to class in the morning." And if I were alone, I could not have done this. But I was with all these other people who were doing it too. And we just had fun staying up at nights, and we had fun doing this. And I came out of that with two major things. I had a love of embryology. I did not know that embryology was a science. And I certainly didn't know that it had anything to do with DNA. And I also came away with a respect for the history of science. Again, when people talk about history, they usually don't talk about the history of science. They talk about the history of battles and the history of politics. And I learned for the first time that there were people who actually wrote about the changes of ideas that happen and how these ideas change from one century to the next. And so that was a very important summer for me. And so when I went to college, I already kind of had the receptors for embryology and history of science. And so I had, those two were very early mentors, Jean Cook and Michael Summers, very early mentors. Then I had mentors, but the last mentor was probably the most important for me. He was my postdoc advisor, and he lived the life that I wanted to live. He lived a life of science. He lived a life of music, and he lived a family life. he spent at least an hour a day having lunch with his wife. He was a world-class scientist, but he put the science into a larger context. And that, for me, was critical to see that somebody could do this, because mostly what I saw were people who did science, and that's all they did. And they wanted to be the first with the best. They wanted to be known for their science. And here was a person who wanted to be known for doing good science, but he also played the piano beautifully. He also had a family and was concerned about his children. I mean, he was a whole person doing science and doing good science. And this person, Robert Auerbach, was kind of the mentor. He never mentored me directly. He just was an example of who I might want to be. And he was also a great teacher. He gave great lectures. And so I try to teach kind of in his image, the way he did.
Since you already brought this up, I would really like to ask you, like, what is your mantra of maintaining a work-life balance? Like, how do you do it?
Yeah, two things. One is that for maintaining a balance between the laboratory and married life, never consider yourself in the first person singular. It's always a we. It's always an us. And just kind of get rid of the I pronoun. It doesn't exist. In the laboratory, it can exist, but even then you're part of a team. And like a holobiont, like we're teams. And so I find that, one incredibly important thing is thinking of myself as part of a team, as a we rather than an I. The best advice I got on teaching, I got from Leonard Bernstein, the music conductor, when he gave a talk. And someone asked him why he conducted so much Mahler for the New York Philharmonic. And he said, I love Mahler and I love my audience. So I want my audience to love Mahler as much as I do. And so I love developmental biology, and I want my students to love developmental biology as much as I do. And so it's a love relationship, multi-part love relationship going on. And I think that's the best way to teach, because a lot of the facts that I'm giving my students will be outdated by the time they are grad students. So I have to give them a kind of a way of thinking or an excitement. I think many students are looking for a tradition, something that they could find themselves grounded in. And the tradition of looking at developmental biology and asking the questions. They're the same questions that Aristotle asked. They're the same questions that, Aquinas asked, that, Harvey asked. People, the first cognizant hominid looked at his baby and said, how did this happen? Why are there 5 fingers? And they're the same. I just figure that these are the big questions, and that's how can you not be interested in finding out answers to these?
You mentioned about history of science, and actually a lot of us and how, like, we're never taught the history of science, as you're correctly saying.
Mhm.
And so I had been reading this book of late, which was by The Power of Infinity by Stephen Strogatz. And it basically introduces you to the history of mathematics and how did, like, Archimedes first thought of pi,
Right.
and it's just so wonderful. Like, it's in the form of that, how intuitive math really is. Whereas, in our schools, you just teach the formulas and then you do problems, and people often develop this phobia. But when
Yep.
you go to the history, it's just so fascinating. And it also reminded me when I was studying this book called Molecular Biology by
Mhm.
Weaver.
Uh,
And in that book, it's like it is so much in detail on how each idea developed and how these things came about, and it is so fascinating when I first read that book.
yeah.
Yeah.
Well, yeah. The opposite of the history of infinity is the history of zero,
Yeah.
Yeah,
which comes from India, I believe.
Right, right.
absolutely. Yeah. Yeah.
Yeah.
So recently, I was just trying to find out books that actually talk about history of science. Like I read Unraveling the Double Helix,
Uh-huh.
and I was so excited after reading that book
Mhm.
that like I wanted to read more of that. So I just thought more of that in Indian context. Like how did Indian science progress?
Mhm.
Since like I feel that it's very important to know my roots.
Yep.
So
Now,
so
have you ever seen the International Journal of Developmental Biology special edition on India?
No,
no,
Okay.
I
not
haven't.
really.
Okay.
No.
It's online. It's free. Uh,
Okay.
Oh,
and
I'll look
it's
it up.
the International Journal of Developmental Biology. And they had a special issue. It might have actually been two issues. Oh, maybe 10, 20 years ago on the history of developmental biology in India.
Oh,
Oh,
wow.
Yeah,
I have to check that
yeah,
out then.
yeah. The
Yeah,
first part was the history, and the second part was contemporary laboratories. And the editor in chief was uh, Vidya Nanjundia in Bangalore at the Indian Academy of Sciences.
Yeah, I think he was in ISC.
Okay,
yeah. I
ISC
see.
Bangalore, yeah.
but
Yeah.
uh, but I think he was the general editor of it, and
Okay.
it's an excellent compilation of all the different parts of Indian developmental biology, and it's just so rich.
Oh wow. I'll check that out.
Yeah, I think you'll enjoy that.
Yeah.
So, all my foundational knowledge of developmental biology has been gained through reading your book on developmental biology. I'm sure it is a book that is very close to the heart of every developmental biology student. So, what is the story behind writing this revolutionary book of developmental biology?
When people when people ask about kind of my writing the book, I have to say that I I feel that I am a surfer on the beach, and I caught the perfect wave. Uh, Now, to be a good surfer and catch the perfect wave, you have to have the skill set to ride the big wave. And there are
Yeah.
lots of people have those skill sets. Not a whole lot, but I had those skill sets. But to catch the perfect wave, you have to be at the right place, and there has to be a wave. And and so you have to have the skills, and you have to be at the right place at the right time. And I was so lucky. Uh, I was at the University of Wisconsin as a graduate student, and I had just accepted a job at Swarthmore College to teach developmental biology. So I looked to see what books would I teach from, and I don't like any of the books. There were the books that were endoderm, ectoderm, and mesoderm. And then the last chapter was genes play a role somehow. And then there was DNA, RNA, and protein. And somehow they make the ectoderm, endoderm, and mesoderm. but there was nothing that combined the two. And I was frustrated. And Andrew Sinauer, who was the publisher, was looking for someone to write a textbook in developmental biology that would combine classical and molecular biology. And he was going around to different universities. He wasn't looking for me. He wanted David Sonneborn at the University of Wisconsin to write the book. And he spoke to David, and David said, I have a good grant. I have great students. I have a project I need to get done. I don't have time to write a textbook. But there's this kid down the hall, this postdoc, who's been griping about not having a textbook to teach from. Maybe he could write one. And so I got a phone call to go down to Dave Sonnenbund's office, and Dave introduced me to Andy Sinauer, and we spoke for a while. And he said, look, you have a background in developmental biology, which is both cellular and classical. You've done gene cloning, you know the molecular techniques, and you have a background in history of embryology. So this is where that history thing comes in very handy. He said, you could write, why don't you try writing 4 chapters? Send me 4 chapters. I'll send them out for review, and then we'll see what happens. And so I wrote the 4 chapters. I was thrilled to write these things down because I was hoping to lecture on these when I had my new job. And I was camping in British Columbia in Canada, and I called my parents to let them know that I'm okay. And my mother says, by the way, I just heard from this guy, Andy Sinauer, who says he wants you to publish a book with him. So that was how I found out. And so what, I was a postdoc. You know, nobody knew who I was. I had the good fortune of working in some excellent laboratories, but I hadn't published much in developmental biology. But I was given this opportunity, and I was going to teach developmental biology at a liberal arts college. Now, Swarthmore College is a liberal arts college, which means there is one developmental biologist. There is one geneticist. There is one neurobiologist, one animal behavior person. So I'm the only developmental biologist there, which means I have to teach the whole course. And so this was a great place to be. And when I was writing the book, the first wave happened. And the first wave was the molecularization of developmental biology. When the first edition of my textbook has no transcription factors, it has no paracrine factors, it has no signal transduction cascades. That happened. The first part of the molecularization of biology in Drosophila and Xenopus came in 1984 as I was finishing the book. And so I put those things into the book, the New Coop Center and the Easter morphogen. I put those into the book. And then the next edition had transcription factors, and the next edition had paracrine factors. So the short answer is we did not know much about developmental biology. And so if you look at the first edition of the book, it has big pictures, big margins, because we didn't know much.
While you were saying this, I just realized that, you know, a lot of professors create these notes and later they just like give the handouts to students in the class. So you actually gave the handout to the world.
Oh, thank you. That's correct. And actually the class that I had in 1984 and 85, they got the preprints of the chapters to read.
Wow.
And these students told me whether or not they felt that it was written well, that was there ambiguity, was I making myself clear? So the students in my class got to actually change some of the things in the book. It was very interesting because, as I said, I was at the right place at the right time. And then there was lateral inhibition. There were other people who said, hey, I have to write a book because there's nothing I could teach with. But when my book came out, they said, oh, I don't need to anymore. So I think my book inhibited some people from writing books because it was close enough to what they wanted. Uh, but so we were lucky in many respects. So the first wave was the molecularization of developmental biology. The second wave was Evo Devo, and I was pre-adapted for that. I was accepted for Evo Devo because I had taken courses in the history of embryology, and I knew the questions that were put to the side when genetics came into the field, and genetics took over evolutionary biology. I knew the arguments that Darwin was having with Haeckel and Owen and Huxley, and those same arguments were now we could go back to with molecular data. And so I was kind of ready for that wave. And so I was just very lucky to have the tools, the toolkit, and be at the right place at the right time.
The progression of science has changed massively in today's world. There are several challenges that one has to go through to become a scientist. What are the challenges you faced in your times to become a scientist? And what do you think, like, how is it different from today?
I think it's very different from today, because today I think there's a lot more emphasis placed on applicable science, and science that's going to make someone some money. I think when I went into science, no one was the CEO, no scientist was the CEO of any company. No scientists had patent rights. Those were industrial scientists, and they were not taken seriously. Now it's the opposite. Now, if you're a good scientist, you're expected to be a CEO of a company. You're expected to have patents.
Do you think that it affects the growth of the real basic science?
I think that it takes funds away from certain sciences and gives them to other sciences. I think that it's harder to get grants in basic research. And when I was starting in science, basic research, people wanted to fund basic research because they felt that there weren't enough scientists. And so it was, so in that sense, it was a very different world that I was starting in.
That there are several failed experiments that we go through, or like sometimes it just doesn't work with the student and the mentor. Have it ever came into your mind that, I'll just leave science, this is not working out for me?
Yes, I have had that experience, and I had the experience, what am I going to do with my life if I don't do science? My parents wanted me to be a musician. And that was always in the background. But I thought, oh, maybe I'd own a bookstore. I'm running a bookstore with a coffee shop in it. I mean,
yeah,
I have had those thoughts. Yes. Yeah. When I was a grad student, but also when I was an assistant professor, and the research was not working, and the students, working at a liberal arts college, I don't have graduate students. I work only with undergraduates. My research is done only with undergraduates. And as you know, undergraduates are taking organic chemistry. They're falling in love. They are finding a political cause. They're trying to find the meaning of life. They have other things on the mind besides my laboratory. And I have to recognize that this is the case. And so I found that my experiments were not working. I didn't have time to do them because I had so much teaching responsibilities. And I just thought, I don't know how I'm going to survive. Fortunately, some things worked out really well. I got a grant to go to Helsinki. And when I went there, I learned new techniques that I could bring back and use in the laboratory. And that was very good. Originally, I tried to have a mouse colony in an undergraduate teaching institution. And that was so, so impossible.
So you
Wow.
have experienced both the worlds of being a researcher as well as a book writer. What are the
Yes,
habits one should develop if they want to become a scientist versus becoming a book writer?
I think one needs a good sense of humor. I think one has to realize, hey, I'm being asked to do impossible things. I think one has to find the enjoyments of both, the enjoyments of writing, the enjoyments of teaching, the enjoyment of research, and not feel badly when one isn't working. Because the trouble is, when something isn't working, you say, oh, why am I doing this research? I could be writing. Why am I doing this writing? I could be getting some research done. And this, by the way, I find to be a very challenging thing, to take enjoyments in what one is doing and not feel guilty when it's not happening. This, and I don't want to be patronizing, but the women who go into research have a big problem if they have children. And if the research isn't working, why aren't I with my children? And when the children are being bad, oh, heck, I could be doing so much more research. And one has to be able to enjoy both and not feel guilty when one isn't working. It's a very difficult thing to do. When I was teaching and doing research and writing, and one of the things wasn't working, I always was feeling, oh, come on, I should be doing the other things, or I should be with my kids, or one can't feel badly, because most of the time the experiments are not going to work.
That's the truth of graduate research life,
my
graduate student life.
my daughter is an artist. She's a glass blower. And I'm always amazed when I see some of the things she's making. But she tells me that the one that I see is like the one out of 50 that survive her criticisms. Most of them get put in the garbage and recycled. And she told me that she thinks of each object of glass she makes as an experiment. She's going to try to make something new. And
Right.
most of the time, it doesn't work. I said, I know this feeling.
So like your parents wanted you to become a musician.
Yes.
I wanted to become a musician on the first hand. And like I was trained in Indian classical music for like 12 years.
Oh,
And
wow.
the crazy part was that so I have changed several teachers in like these 12 years, 12 to 14 years. I'm still like, when I came back home again to reapply for places for PhD, I actually started practicing music again, because I thought that was the most exciting thing in my life for a very long time. And my music teacher actually used to explain the science behind the music instruments. So like, and then he used to he used to say different pitches, and then he would say that you have to experiment with your voice if you want to uh, if you want to sound different, you have to experiment continuously with your voice.
Mm-hmm.
And I think that's where experimentation started in my life.
Interesting. Well, I have written somewhere that developmental biology can be considered embodied music, and people describe differentiation as a symphony, that that you have all these genes being coordinated together. It's actually, I think probably the closest thing I could get to what the music is like a Javanese gamelan, where there's no conductor. It's it's an orchestra, but they hear a thing and then they play their note. I think that a lot of developmental biology its patterns, its rhythms, its syncopations. There's a lot of music going on in developmental biology.
True.
For sure.
Absolutely.
There are several debates and issues raised regarding open science publication, especially when it comes to the amount that is charged to publish in open science access journals. What
Yes.
is your opinion on open science publication?
I think that open science publication should be stratified by need. And in some cases it is. For instance, I teach at a liberal arts college. I do not have anything in my grant to publish in this journal, in Frontiers, for instance. But the people at Frontiers have a system that said, okay, we have asked you for an article. If you do not have this in your grant, you do not, the fee is waived.
Yeah.
And I think that it should be stratified by one's grant and by one's university. The people at Harvard should have to pay. The people at Berkeley should have to pay. And part of what they pay should be so that others don't have to pay.
Yeah.
And it should be based on if the report, if the science is good enough for the journal, how do we get it into the journal? And I think that again, open access is a double-edged sword. I really feel that authors should, authors like of textbooks should be paid for what they do. Publishers should be paid for what they do. I think that you should be paid for intellectual achievements, intellectual work. But I think also that that work has to be disseminated to whoever wants it. And so the publishers have to charge enormous amounts of money, but I don't think they have to charge enormous amounts of money to everybody. And and I learned something when I was on my sabbatical leave in Helsinki, which was very interesting. The head of my laboratory in Helsinki came back very angry. And I said, what's the problem? And he said, I got caught speeding in a residential area. I said, well, how much did you have to pay? And he said something like 500 euros. I said, that's a lot of money to pay for a traffic ticket. How fast were you going? And I said, about 5 kilometers over the speed limit. $500? Okay. He then said something interesting. He said, that's because I make a lot of money. It would have cost the technician only $30 for the same offense. So they don't put a price tag on a crime. They make it the same amount of grief. It's a graduated percentage of your income. So that if you are very wealthy, you pay a lot more for the same crime as somebody who is relatively poor.
So coming to social media has both like its pros and cons. How has it impacted you?
Minimally, because I am addicted to email. I know that if I had Facebook, it would just be like a conduit, I would just be looking at it all the time. So I don't have Twitter. I don't have Facebook. When I got a co-author for my textbook, my co-author is very savvy with electronic media. I am not. I really am very old-fashioned when it comes to electronic media. And so I leave that to my co-author, to my publisher. I stay away from it.
So if you were given the power to change something, what would be that one thing that you would want to change about academia the most?
I would want to give professors more autonomy to teach what they want to teach the way they want to teach it. I don't like people being told this is what you're expected to teach. This is what we want you to, the people to know. And that, I would say, would go down into the high schools. I would want high school teachers to be able to be creative in what they teach and to have you're expected to teach, development, cell biology, evolution. And here are, maybe 3 things in each group that, you have to make sure that the students know. But how you teach it, the examples you use, we're not going to tell you. You have to make your own syllabus and be creative in it. The other thing would be to work together across disciplines more easily, to have more grants, more funding, so that a biologist could work with a person in music theory or in, religion or in poetry, so that you get to experience the other parts of the world, because I think it makes life better, but also I think enriches teaching. A friend of mine, Bob Root Bernstein, has said that the best education for young scientists is artwork. And whether it's music, dance, sculpture, painting, the ability to recognize forms, patterns, rhythms, the ability to use your hands, to use your fingers. There's so much lab work, which, if you were in the arts, you would know how to do. It just comes naturally.
Yeah.
And so I think that, more hands-on things and discussions between people in other disciplines, I think is really critical. And this is one of the advantages I had at a liberal arts college. Most of my friends were not in the biology department. There were only 10 of us biologists. Most of my friends were in music, economics, psychology, religion, English literature. I found out interesting things when I worked in Finland, and Finland supposedly has the best education system in the high schools in the world. The reason they have this, the reason they have the best situation for their high school teachers and for their students, is that it is very difficult to become a teacher in Finland. It's harder to become a teacher than it is to become a physician. That when I was there, there were only so many schools that taught teaching, and there were more schools that taught medicine. And so becoming a teacher was difficult, and you had to have high grades to get into a teaching school. In America, low grades will get you into a teaching school. Then what was amazing is in the teaching schools, when you're at a university taking courses in education, you can fail out. It isn't once you're in, you stay in. No, you could actually fail out if you're not doing well.
Wow.
And we had people in our laboratory in Helsinki who I thought were PhD students. They weren't. They were masters in education. But to get your master's in education, you have to get a master's in the field that you might teach in, whether it's mathematics or literature.
Wow.
And so these people, and once these students get their teaching degree, the government of Finland says, okay, you know how to work. You did this very difficult job. We respect you. And we are going to give you a high salary position with respect. And part of that respect is you could teach the way you want to teach. And
beautiful.
And as a result, the teachers are excellent and are still excited about what they do. And, but it's a matter of getting respect for teachers. I think that's absolutely critical.
You are actively involved in science communication. So what changes have you seen over the time in the pattern of science communication?
Oh boy, yeah. The internet has revolutionized science communication. The digital photography has revolutionized embryology education. The ability to show movies in PowerPoints has totally changed the way I teach embryology both to my students and to public audiences. I think it's easier to get people excited when they see a salamander embryo developing in 1 minute, from egg to larva. It gets people fascinated when they see the egg dividing and the neurula forming and stuff. And that has changed. It's made our job easier to a degree I never thought possible, because we can now show beautiful photographs and incredible movies. So I think that in terms of public education, the things that I'm seeing on television, the nature documentaries, are just beautiful. I think that the technology of education has been amazingly good, and it's improved. I think education is actually improved, both for the, it's there. Whether people want to see it or not is something else. Again, when I was growing up, the television was full of educational programs. Most of the television was full of garbage, but there were educational programs if you wanted to see them. And I think that now there, again, are really good educational programs, but they're only on certain channels. They're not on the main channels, but they're there, and YouTube has made so much available. So in addition to all sorts of really awful misinformation getting out there through the channels, there's also just remarkably beautiful things for embryology and developmental biology, which is just absolutely astounding. When I look for things, when I was looking for things for class, I would go on the web and I'd find these YouTube things made by scientists and made by educators, and it's amazing.
What would be the one advice that you would like to tell all your young audience?
Retain your love for the world, for your discipline, for your family, and have a good sense of humor and a sense of toleration. I mean, I don't know, I think there's not one thing that I could say, the Dalai Lama said, be kind to people, and be kind to yourself. That goes a long way.
Coming to our last question, what do you think about Kitchen Lab?
Oh, I think it's a great idea. I think the more people who find out about the joys of doing science, and that anyone can do science, that it is not for an elite, it's science has always been done by people. They haven't always called themselves scientists, but that science is one of the things that separates, humans from most of the other animals. We have an ability to experiment, whether or not we do it formally or informally. All you have to do is look at a 5-year-old. They are constantly doing science. And we just have to keep that 5-year-old in us. We have to keep the excitement of learning new things and discovering new things, because we still don't know the basics. We still don't know the basics of development. So, I just think that we can keep the child in us, that everyone is a scientist. Everyone was born a scientist. And if culture hasn't hammered it out of us, some of the few can actually call themselves scientists, but everyone does science.
That was so great. And thanks a lot for joining us. It was amazing talking to you.
Best wishes to all of you in this group. Congratulations on kitchen science, and really, I hope that you bring a lot of people into what we call science. But even if they don't go into science, just a way of appreciating what they do in the kitchen from day to day.